Hello students. The topic for discussion today is preservation methods for non-chordates. The preservation methods for chordates I have already discussed in a separate PPT, and now in this present PPT I will going to discuss about the preservation method for non-chordates. So I am Dr. Bhagyashree Mohanto, Assistant Professor, Department of Zoology, USTM. So before preservation, fixation is a very essential step. So what is fixation? The fixation of biological specimens involves the coagulation of cell contents into insoluble substances with the purpose to prevent autolysis and the degradation of the tissue. So before preservation, fixation prevent the specimen from autolysis and degradation of their tissues. Fixation coagulates and stabilizes protein in specimens so that they do not distort or deteriorate during preservation, study and storage. So without fixation, we could not preserve a specimen, or if we preserve those specimens without fixation, it is very difficult to study them and to store them in our museum. A good fixation is generally achieved in a brief amount of time and as soon as the animal is collected. So when we collect our specimen after the same hour or day, we have to fix those specimen to avoid deterioration or degradation of the tissue. Generally, the fixative we have used to fix the invertebrate species is 4% formaldehyde solution. After fixation of invertebrate species, we have to preserve those species. So preservation means it is a process that serves to keep the dead body of an organism from decay in part or in whole, presumably to be studied later. So both vertebrates and invertebrates can be preserved in fluid or as dry specimen. So we have two types of preservation technique. One is dry preservation and other is wet preservation. The archival preservation fluid that has been used to longest and is generally preferred is alcohol. Then the standard is 70 to 75% ethyl alcohol or ethanol. 40 to 50% isopropyl alcohol is also used on some animal taxa. Sorting solution is also used to preserve, and it is made up of 1.5 parts propylene phenoxetol, 5.0 part propylene glycol, 10.0 part full strength formaldehyde and 83.5 parts of distilled water. So this aggregation of chemicals is called a sorting solution, and it has been used successfully for long-term storage of some particular taxa of invertebrate. So before fixation and preservation of an invertebrate species, killing is must, because if we do not kill properly, the invertebrate species will be shrink or sometimes distorted. So therefore killing method is a very essential method before fixation and preservation. For killing method of invertebrates include killing jars. So, liquid killing agents are like ethyl acetate, ether, chloroform and ammonia are used in some jars. And ethyl acetate is most widely used chemical. All of these chemicals are extremely volatile and flammable and should never be used near fire. So sometimes we use solid killing agents except these liquid killing agents. So solid killing agents are basically cyanides like potassium cyanides, sodium cyanides, calcium cyanides, etc. They are very dangerous, rapid acting poisons with no known antidote and hence are to be handled with extreme care. So for solid killing agents, we actually use the cyanide materials and we keep them inside a jar, and in that jar we put our invertebrate for killing. So in these killing jars, we sometimes have to use absorbent materials like plaster of Paris or cotton for to avoid the deterioration of the outer surface of invertebrate species. So killing jars contains very dangerous chemicals either in solid form or in liquid form. It is a very deteriorating effect on environment. So for the safety concern of environment, sometimes we use another kind of killing method that is called as freezing. So after the specimen have been collected, they can be transported home or to the lab in a plastic Ziploc bag or paper envelope. Then the specimen are carefully placed into a portion of the freezer where they will not be damaged. Living invertebrates in the freezer or prolonged periods of time, however, may damage the specimen. So we should keep our invertebrate specimen inside those freezer for a very short period. So they are to be freezed only long enough to render them immobile. So with this process, we can kill some of our invertebrate specimen. Now let us discuss about our main topic, that is preservation method of invertebrate, and first we will going to discuss about dry preservation method. So there are lots of invertebrates in our world, and so for different invertebrate species the dry methods are different. For first I will going to discuss about sponges, how to preserve the sponges in dry method. So before preserving the sponges, we have to wear hand gloves. Then we have to put the sponges under clear running water, rinse and squeeze to drain out excess water. Then for removing the smell from the sponges, we have to place them in a container of alcohol with a lid for 48 hours. Then, before drying large specimen, labels are attached by means of string threaded through the body of the sponge. Then we need to dry it in the sun. When thoroughly dried, sponges are kept in small cardboard boxes supported with tissue paper. Then naphthalene flakes may be added to the dry container to avoid deterioration. So this is the dry method of preservation of sponges. To preserve stony corals in dry method, first, the animals using the coral as a home must be removed, and this is achieved by soaking the coral in fresh water, because corals are saltwater animals and they could not be survived in fresh water. So we need to soak them in fresh water till they die. And after they die, we can remove the coral out of the animal. Then the corals are to be soaked in 2 parts of bleach and 1 part of water for 48 days. And for preserving the coral specimen, we need to soak the coral in alcohol and let it dry and preserve in the museum. To preserve horny corals, the specimens are dipped in neutral formalin for 15 minutes and then dried in a warm but shaded place. Prior to drying, branches are so arranged that they will take up the least amount of space in a museum tray. After the specimens are thoroughly dried up, they are kept in light-proof boxes with a few crystals of para-dichlorobenzene or naphthalene. So, example of horny coral is gorgonia you can see in the picture, and you can see in our museum also. To preserve dry shells of mollusk, the first step is to be clear the shell of any dead tissue which is a cause of bad odor. Most shells can be cleaned by coating them on the inside and outside or filling it completely with a dry solution, like 3 parts of salt, 1 part of baking soda, and freezing them in a zipper bag. An alternative to the process of cleaning dead tissue is to boil the shell. After removing the dead tissue, the shells are sorted out into sturdy and fragile shells. Two sturdy shells are soaked in container having 4 cups of water mixed with 4 cups of bleach and 1 tablespoon of dishwashing liquid. They are periodically scrubbed with a toothbrush to remove any seaweed, algae, branches, and debris attached to them. Fragile and bivalve seashells are washed in mild dish detergent. After the seashell is clean, it is removed from the solution, rinsed well, and dried gently with paper towel, and they are then air dried or sun dried. Some collectors like to use baby oil on a well dried shell to bring out its luster. For echinoderms dry preservation, we need to clean the animal in a mild detergent solution, that is mostly water and a very little detergent, and dry it in the sun. We can soak the starfish immediately in rubbing alcohol for 48 hours and forego cleaning it. It is then allowed to dry in the sun. We can also soak it in formalin, which is 1 part formaldehyde and 5 parts water, and then dry it properly. While drying, it is necessary to put weight on every arm of the starfish, or else they will curl up. To preserve insect, we need to first relax them. The relaxing method includes flat plastic container with an airtight lid makes an ideal relaxing dish. Baseline with moist cotton wool or synthetic sponge saturated with water and covered with a blotting paper. Phenol crystal, few drops of Dettol added to prevent fungal growth. A sheet of blotting paper is also placed on the inside of lid to avoid condensed water to fall over the specimen. For preservation of invertebrate specimens like insects in dry method, we have to add some particular pins, and these pins are basically 3 types. First one is English pin. The English pins are 18 to 30 mm long and quite stout. These can be handled well with pinning forces and preferred for staging or double mounting. We have continental pins. Continental pins come in 3 sizes, 35 mm long and 38 mm long, and finally 50 mm long. These are good for direct pinning. We have minuten niden. Minuten nidens are a very fine and short pin, having 10, 15, 20 or 22 mm, and with or without heads, and used for only double mounting or very small insects. For mounting insects, we need to use different types of equipments. First one is pinning block. It is a very important equipment because specimens and labels are placed in a uniform height on the pins, and to achieve it we need to use this pinning box. Then setting and spreading for larger insects like moths, butterflies, dragonflies, grasshoppers, cockroaches. We need to spread and set those specimens with their wing in a spread condition. So moth, butterflies, lace wings and dragonflies set with both pairs of wings spread, and grasshopper, cockroaches, mantids, stick insects and occasionally bees are set with only one pairs of wing extended. For mounting of large insect over 8 mm, we use direct cleaning method. The body part through which the pin is placed differs in some orders. Here in the diagrams you can see how we can mount large insect with pins. So let us discuss about preservation method of invertebrate, that is wet method. So in wet method we use certain amounts, certain types of fixative and preservative chemicals, and with the help of them we can preserve them in our museum. So first we will discuss about porifera. For poriferan species, anesthetizing is not necessary. Fixative we have used, that is 2 changes of 50% alcohol, and 70% alcohol is used as preservative. For Cnidarian and Ctenophora, for anesthetizing, few crystals of methanol added to water in which animals have been allowed to extend. Then after anesthetizing, we can fix those specimen in 20% buffer formalin, and 10% formalin or 70% alcohols can be used as preservative. For platyhelminthes, for anesthetizing, we can use hot alcohol. As a fixative, we can use 70 to 90% alcohol, and as preservatives, we can use 70 to 90% alcohol solution as well. For nematodes, anesthetizing is not necessary. For fixative, we can be used 3 to 5% hot formalin solution or hot 70 to 90% alcohol, and to preserve those specimens we can use 10% formalin or 70% alcohol. For nematophora, anesthetizing materials are not needed, and we can directly fix them in 3 to 5% formalin, and 3 to 5% formalin is also used as a preservative. For bryozoa, for anesthetizing, slow addition of methanol crystals to water in which animals have been allowed to extend. Then rapid addition of commercial formalin or buin solution is used for fixation of that species, and after fixation 5% formalin solution or 70 to 90% alcohol can be used as a preservative. For Annelida, for anesthetizing 5 to 10% alcohol for 10 to 15 minutes we have to use. Then after anesthetizing, fixative 4% formalin as a fixative we can use, and to preserve those specimen we can use 70 to 90% alcohol. For arthropoda, here anesthetizing is not needed. We can fix the arthropoda species in freezer or in killing jar or 70 to 90% alcohol, and to preserve them for non-insects we can use 70 to 90% alcohol, and for insects drying and pinning methods are there. Then to preserve mollusk, methanol crystals to water is used as anesthetizing agent. To fix the marine mollusk, we can use 5% neutral formalin, and for freshwater and terrestrial mollusk, we can use 70% alcohol. In preservation, for marine, 5% neutral formalin, and for freshwater and terrestrial, 70% alcohol. Like in fixation, we can use. To preserve echinodermata, first we can use methanol or magnesium sulfate crystals in water as an anesthetizing agent. Then for fixing them, we can use 10 to 12% buffer formalin. And to preserve them, we can use 70 to 90% alcohol, and some can be dried. The preservation method for parasitic invertebrates are different from other invertebrates. So let us discuss about the methods. Arthropod invertebrates preserved in 70% alcohol in glass vials with screw caps lined with anti-evaporation inserts made of plastics. Mite, ticks, lice, fleas, houseflies and bugs can be dropped straight into the alcohol. Nest fly larvae should be killed by immersion in boiling water prior to storage in alcohol, or better yet, immersed for 24 hours in KAAD solution. KAAD solution means 1 part kerosene, 10 part 95% alcohol, 2 parts acetic acid and 14 parts of dioxane. If specimens are to be stored in alcohol for long times, that is for years, 5 to 10% glycerin should be added to the alcohol to prevent hardening of specimen. To prevent small vials from drying out in long-term storage, they can be placed in an inverted position between layers of cotton in a larger jar of alcohol. So long as the caps are not loosened in this process, this ensures that the fluid in the vials will be the last to evaporate should they go unchecked. If specimens are to be mounted in slides, they should first be washed several times with 70% alcohol to remove the glycerol. So these are some of the preservation technique of different kinds of invertebrate parasites. Dear students, that's all for today. To prepare this lecture, I have gone through two books, that is Animal Taxonomy Manidev Raj and Theory and Practice of Animal Taxonomy and Biodiversity, written by V.C. Kapoor. And hopefully my lecture will help you in your studies. So thank you very much for today.